Question number 161. Select the odd one with respect to direct gene transfer in plant protoplasts. So the keywords are direct gene transfer and in plant protoplasts. And the options are electroporation, disarmed TI plasmid, microinjection and particle gun. And the correct answer to this question is option number 2, disarmed TI plasmid. The methods of gene transfer involves direct and indirect methods. Direct methods of gene transfer and indirect method of gene transfer. Direct method of gene transfer do not involve vector, whereas indirect method of gene transfer involves use of vectors. So we have to find out. So disarmed type plasmid is a plasmid. It's a vector. So it is a type of indirect method of gene transfer in plant protoplast. Whereas rest of the options, electroporation, microinjection and particle gun are the incorrect option because these are direct method of gene transfer. In electroporation, short electrical impulses of high field strength are given to the target host cells. So this is a direct gene transfer method. In microinjection, recombinant DNA is directly inserted into the nucleus of animal cell. So this is also a direct gene transfer method. In particle gun, or also known as ballistic or gene gun method, cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA. So these are the direct methods and the incorrect options. And the correct answer to this question is option number 2. Let's proceed to the next question. Question number 162. Downstream processing of desired product does not involve. And the options are isolation of product, purification of expressed protein, marketing of extracted product, and expression of foreign gene. And the correct answer to this question is option number 4, expression of foreign gene. Expression of foreign gene is a step in upstream processing and not in downstream processing. Downstream processing involves isolation, purification, formulation, packaging, storage, and marketing of products, so option number 1, isolation of product is an incorrect option. Option number 2, purification of expressed protein is an incorrect option. And option number 3, marketing of extracted product is also an incorrect option because these are the steps which are involved in downstream processing. Whereas the first option, expression of foreign gene, is a step of biosynthesis. So the correct answer to this question is option number 4. Let's proceed to the next question. Question number 163. Choose the option that represents an incorrect statement with respect to bioreactors. And the very first statement given is also known as fermenters, true, for growth of microbes under a controlled environment. And as it is a correct statement, so this cannot be a correct answer to our question because bioreactors, which are also known as fermenters, they are used for the growth of microbes under a controlled environment. The next statement is large volumes, that is 100 to 1000 liter of culture can be processed. And as it is also a correct statement, so this cannot be a correct answer to our question. Next statement is anti-forming agents are required to increase the forming caused by presence of proteins in the culture medium. And as it is an incorrect statement, so this is a correct answer to our question because anti-forming agents are required to reduce the forming caused by presence of proteins in culture medium and not to increase the forming. The next statement is bubbles dramatically increase the oxygen transfer area, and as it is also a correct statement with respect to bioreactor, so this cannot be a correct answer to our question. As you can see here, these bubbles, they increase surface area for oxygen transfer. So the correct answer to this question is option number 3. Let's proceed to the next question. Question number 164. Read the following statements and choose the correct option. In statement A given is, in batch fermentation, the conditions once set are not changed from outside after the process of fermentation starts. And this is a correct statement because batch fermentation is regarded as closed culture system. And so this is a correct statement. It is in continuous culture system in which the used medium is drained from one side while the fresh medium is added from another side to maintain the cells in their physiological or most active exponential phase. In batch culture, the conditions once set, the initial conditions, they remain unchanged. They are not changed from outside. So it is a closed culture system. Statement B is, batch fermentation is called an open culture system, and this is an incorrect statement because it is a closed culture system, whereas continuous culture system is an open culture system. Now let's discuss the options. The first option is statement A is correct, and as it is a correct option, so this is a correct answer to our question. Option 2, statement B is correct, is an incorrect option. Option 3, statement B is correct explanation of statement A, and this is an incorrect option. And the last option is both statements are correct, and this is also an incorrect option because statement A is correct but statement B is incorrect. So the correct answer is option number 1. Let's discuss our next question. Question number 165. In simple stirred tank type of bioreactor, stirrer is responsible for, and the options are increase in the pH, decrease in temperature, extraction of product, and uniform availability of oxygen. And the correct answer to this question is option number 4, uniform availability of oxygen, because a stirrer or agitator is responsible, or stirrer facilitates uniform availability of nutrients and oxygen in bioreactor, whereas the other 3 options, increase in the pH, decrease in temperature, and extraction of product, are the incorrect options. Because this simple stirrer type of bioreactor has a separate agitator system to facilitate uniform availability of oxygen. So this is correct. It has a separate pH controller system. So this is an incorrect option. It has a separate temperature control system. So second option is incorrect. And separate sample ports. So third option is also incorrect. So the correct answer is option number 4. Let's discuss our next question.